Good morning, everybody. Welcome to our last day of the conference. I'm delighted to welcome you this morning, but also welcome our third keynote, Lionel Guidi from Sorbonne University in Paris. He's also an affiliate at the University of Hawaii, but also he's a CNRS researcher in France, Visual France de la Soumer. And today we're very happy to have him here because he's going to be talking to us about a very, very interesting, but also cutting-edge topic. He's going to be telling us about the planktonic social network of the biological carbon pump. I have read about the carbon pump, and it's something which is very, very important because it's actually how the carbon is actually pumped into the deeper layers of the ocean. But also Lionel has been working under the Tara Ocean Expedition, which again is a very important and one of the largest DNA sequencing efforts that has ever been done before. So we are very lucky to have him today with us. So I'm going to introduce Lionel. Please sit back, relax, pay attention, and enjoy the talk. Thank you very much.
Yep. Thanks. Thanks for the introduction. And thanks for the brave that decided to wake up this morning to come to the last plenary. So like Sylviana said, I'll talk about the plankton social network of the biological carbon pump. And it's mainly based on the research that we've been doing in Tara. So it's me that I'm doing the talk, but actually it's part of a big consortium, the Tara Ocean Consortium. So you'll, depending on where you stand, if you are a computer scientist, a biologist, an oceanographer, or a chemist, maybe you'll see the talk a little bit differently, because the purpose of Tara is actually to do a lot of multidisciplinary science, from computer science to biology. So just to start, a few things about the biological to plankton. Why do we care about plankton? It's because the plankton is part of two main processes, actually. The trophic transfer, a lot of you are probably working on that, and the carbon sequestration. This sequestration is through the biological carbon pump. So I'll talk about this today, but all the concepts that I will present and all the analysis and all this, the way I present the biological pump and also be applied to the trophic transfer. So it's just to give you a flavor of how we think about it. So the biological carbon pump, that's how usually people see it. So that's a main, it's a human view of the biological carbon pump, and it's very simplistic. Usually people try to make good design of it so you can understand it. So you have basically a lot of interaction in the plankton that generates particles, and these particles interact with plankton again, and what is not eaten or not consumed finally sink in the water column and get to the bottom where you have a sequestration of the carbon that is generated. So if you look at just the top layer, the aphotic zone, and what is happening there, you usually try to find the usual suspect, the plankton that matters when you look at the biological carbon pump, and that's what people have been doing for 30 years or more than 40 years. So usually, if you go through the literature, that's what you see. You'll find diatoms that are the usual suspect. It's the big cells that do a lot of primary production. They big, they sink fast. So because they big, they sink fast, they sequester a lot of carbon. But if you browse in the literature, you find also small plankton, like picophytoplankton, because of their trophic transfer, and because of that, they sequester a lot and they might sequester a lot of carbon also. And then it goes on, and it's not just about the species, but it can also be about the way the community is structured, or the function of the species in that community. Now, it's not just the species that matter, but it's also the way the transfer of carbon is done. So that's what you have on the right here. So basically, if you look at the red curve, it indicates how fast the carbon is transferred. And that's critical because it gives you an indication of the remineralization. And what is remineralized doesn't get to the bottom and is not sequestered, but go back to the atmosphere. So people have been working on that, and that's just a diagram that shows you that it's very sensitive. The way the transfer of carbon is done, it's actually can have a huge impact on the way carbon is put back to the atmosphere. And so what you can see here in the gray bar, it's that a slight change in the attenuation of that flux, the transfer, the efficiency. So a small change can have a huge impact of the PCO2 in the atmosphere. So we don't know much about this efficiency and the way carbon is actually getting to the bottom, but we've been doing a few studies, and that's one of that give you an idea of the variation of this value of the attenuation with depth. And basically what we managed to point out is that it's the size of plankton or the community structure that matters, but still this is a very coarse description of the biology that drive the process, that drive the biological pump. So how do we measure the flux? So we usually put sediment trap, that's the big white instrument on the right, to collect particles. The thing with sediment trap, it's been used widely for the last 30 years, but it's very time-consuming. It integrates the flux on large scale and on a long time, and it's hard to deploy, and it doesn't allow you to get a lot of data. So that's an idea of the amount of data that have been collected using this deep sediment trap for the last 30 years. It's a paper from Anjou, and it looks pretty good because, I mean, it's few bars on a small map. But actually, if you think how much information we know about the deep ocean, that's what you have here. It's the picture. So the blue bars indicate everything we know about the deep ocean, of the mesopelagic to the bathypelagic. That's based on a quick literature search. And so in black, it's everything that we know about the moon, research that has been done on the moon itself, or the impact of the moon. And so there is one order of magnitude between the two. So it gives you an idea that basically we know nothing about the deep ocean. And if we know nothing, there is no way we can understand the process. And again, from the first slide, you can tell that what we know about the plankton and the way the plankton interacts with the process, the biological pump, it's a very coarse view of the process, and it's like looking through a keyhole here. We don't know much, and because we don't know much about the plankton, it's the way it interacts, the way it functions, and the deep ocean, it's really hard to get a good grab on the biological pump and to estimate the process at the global scale. So the idea of that was can we do better? And that's why actually I got involved in TARA. It's to study the biological pump with a new perspective, like putting a new view on the process. So the TARA expedition, I'm just going to say a few things about it. So it's mainly, it came out with from the mind of Eric Karsanti. Eric Karsanti is a cell biologist. So why a cell biologist started to study the ocean? It's because for him, like if you take the cell, the way the cell functions, like the system itself, it has a lot. It could be a mirror of the ocean. In system biology, they've been doing a lot of study to try to understand the functioning of the cell, from like the molecule to the cell itself. And it came to the ocean with that view, and that's what we try to do in TARA. But TARA, it's a lot of people. It's 150 people at least, and it's a very multidisciplinary. So we try to put people that have different interests in the same room or here in the same boat to do similar research. So the goal of TARA was actually to get a real link between environmental data, environmental forcing, like climate, pollution, etc., and the way the system, the biological system is functioning. So on the boat, it looked a bit like that. So it's not I mean, for the one of you that are in the room and are going at sea, that's not the usual cruise. TARA, it's a small boat. You're stuck on the back deck, and you're 6 people, and you try to do everything you have to do, from genomics to later on computer science. So we were trying to collect water to do like real oceanographic measurements. We had a very well-equipped CTD rosette. We had a dry lab and a wet lab where we tried to collect the sample and do the first preparation of the sample to send them back to the lab for sequencing, for example. And so that's how we function. And because of that configuration, I think also it helps people to trust each other, to work with each other, and to think about what we want to do with the data. So that's few numbers about the expedition. We've been going around the globe mostly in oligotrophic systems, and then we did a cruise in the Arctic, around the Arctic. It's about 35,000 samples, 200 people, 21 labs, 7 countries. And if you think in terms of cost, it's only 9 million for a 3-4 years expedition. If 9 million include everything, like the cost of the crew, the boats, the science, everything that we've been doing, and the sequencing. So to go back to the sample, the 35,000 sample, so some sample are from the CTD rosette. So this is probably what you are familiar with. So we've been trying to do like classical oceanography, even though the boat was not set up for that at the beginning. But we had a lot of measurement of the environment, like temperature, salinity, oxygen. But we tried to collect also water to do pigment analysis, HPLC, to get the carbonate system, and to get a lot of measurement, optical measurement, to try to characterize the system with optical instrument because it's easy to deploy and to get measurements. So then we had a real focus on the biology, and we tried to collect waters to measure, to see the full biology, almost end-to-end. It's from viruses to large eukaryotes, so it doesn't include fish, even though we had small fish, but it's from virus to large eukaryote. So the way we've been treating the biological sample, it's by omics first. And we got lucky, actually, because the expedition started in 2009, and what you see in that curve is the price of sequencing with time. And what happened in 2008 is that there is a huge drop in the price of sequencing because of new methods like the NGS that came out and made basically the price of sequencing a lot cheaper. If you look at that curve, it's a 4 order of magnitude lost in the price of sequencing in about 5 years. So because of that, we were able to get a lot of sequencing data and play with that. So what we had is about 7,000 data sets of different sources. And we tried to integrate that from metabarcoding to metagenomic, metatranscriptomic, and link that to the environment. And we did a big effort also to try to look at things using different optical and imaging instrumentation. So there is a big challenge here because when you look at the spectrum of life, basically it goes from over like 6 order or 8 order of magnitude in terms of size range. And so you can't use one instrument to do that or to image everything. So we came up with a suite of instruments here that goes from the flow cytometer up to the UVP that gives you a lot of pictures about the plankton from few micron to few centimeters. There's a lot of questions and challenges there because if you want to merge this data set, they come from different instruments, even though it's imaging. The imaging instrument have different properties, and we try to merge all that. So it takes a lot of effort, but at least we have a full resolution of the size spectrum from 1 micron, pretty much, to a few centimeters. So that's the instrument that have been used. So it's the imaging flow cytometry data, the FlowCam, the ZooScan, and the UVP mostly. So what we did is we developed hardware and the software to be able to put all this data together and share it. Because one of the big goals of Tara is to put the data out there so people can use it. I mean, I mentioned the 200 scientists, but actually, if you have 2,000 scientists, it's not enough to treat the number of data, the amount of data that we've been collected for 3 years. So all the data, as soon as they're validated, they put on the web and people can play with it. So if you want to play with it, go on the different websites, download it, and do whatever you want with it. And so for the imaging data, we developed a software because there was no good database for it. So we developed ecotaxa that allow you basically to put imaging and to put annotation on it and to put images with metadata, because having the metadata matters a lot. And so in about 3 years, we have now more than 50 million images, all annotated, but about 50% validated. And so it's the biggest database on imaging now that you have available out there that you can play with, and it's a free resource. So we've been thinking a lot about the data set also, because it's not just like collecting data, it's to be able to allow scientists, other scientists outside of the consortium to work with it. And so it's a big challenge because we have environmental data, remote sensing data, and product from models, imaging data, omics data, and we try to combine everything. So, and there is no one database for that. So what we try to do is to come up with a system to be able to at least get back to the sample if we need to from the data set. And so that's Stéphane Pesan in the consortium that is really working on that. And so everything, like you can see on that plot, is linked. From the sample to the sequencing, to the imaging, to the environmental data, and it's linked to a sample registry that you can download also. And so what we try to do is not just to put the data, but also describe the data set. So now we come up with papers that give you all the information about the data set, its limitation, where you can download it, what you can do with it, etc. So that's a big effort in Tara also, to provide data to the community. And so the way we've been thinking of it is linking different databases that already exist at the French level, the EU level, and now we try to come up with a system that will also allow to link products of this database to the database itself, like the model products. So just few important results from the expedition. That's the first, the main result that probably you heard about. It's about the plankton. So it came out in 2015 in Science as a package of 5 papers. And so the first one is about the eukaryotes. So the first thing that we tried to do is to present the data set and what was new with it. So the first paper is about the eukaryotes. And I just going to highlight a few results about this paper. So we reached saturation at about 1,300,000 OTUs. This is 10 times higher than the number of formally described marine eukaryotes. And about a third cannot be assigned to any known taxa. So after the eukaryotes, we tried to present the prokaryotic data. And again, few results. It's about like 40 million genes. It's 1,000 times equivalent to what has been done in GOS. And if you compare that to human sequencing project, it's about 135 fully sequenced genome, human genome. We also worked on the viruses, and again, few results. So we came up with about 5,500 viral community. Before that paper, only 39 were known. And basically, this data allowed us to support the seed bank hypothesis in viral ecology. And a new paper came out a few months ago, reanalyzing the data, but the full data set, because at the time of the paper, it was only about 36 stations, while now we have 200 stations sequenced and ready for use. And now we came up with about like 15,000 viral communities. So that's, again, it's a resource that everybody can use. And to me, one of the most interesting research that has been done with this data, it's not only presenting the data set and the diversity, but it's linking this diversity. So that's what we call the interactome. It's basically a picture or an abstraction of the community linking species together from virus to eukaryote. So that's what looks like. That's the interactome. So that's, I mean, it's a picture. The thing with network, it's nobody can really understand it, but it's an abstraction. And when you play with an abstraction, you can extract information, and that's why it's very interesting, because it's just a picture of the community. It's a new way of looking at it, and we can play with that. It's not the picture itself, because I mean, getting a nice network helps you to publish in big journals, but it doesn't give you information per se. One information from that network is that biotic interactions were more important than abiotic. And that's critical because it tells you a lot about how much you need to know about the community if you want to understand the process. So now with this paper in mind, I've been trying to think about how we can revisit a process. Here it's the biological pump, but you can think about any other process. So that's a very neat picture of the biological pump. Like I said at the beginning, it's very simplistic. People have been doing that because they need to understand it. I mean, humans need to understand it. And so you try to make nice drawing and get a picture of it. But actually, if you take back to the community, I mean, the plankton, we've been using sequencing, NGS, to try to look at the community. And what it gives you, it's a full picture of the community. And instead of having this, you have this. So here, it gives you an idea about, we have to go from that understanding to that complexity if we want to understand the process. And that's critical. I mean, it's a big challenge. And then about the way flux is being, the way flux is described in the water column. Instead of using sediment traps, we've been doing imaging instrument. We've been using imaging because it's not an ideal measurement of the flux, but it's a proxy of the flux. And when you use imaging or optical instrument, you can get much better resolution in time and space. And so obviously on the boat, it was not possible to deploy sediment trap at all stations. So we came up with imaging instrumentation to try to fill the gaps. So what it gives you is this. And it's a picture of the variation of the flux over the transect. So here it's a bit complex because it's a mix of space and time, because the cruise is 3 years. But at least it gives you an idea of the variation of the flux over the 500 first meters and over the full transect. If you go back to the picture that is on the top, it's what has been collected over 30 years with sediment trap. And in terms of data volume, it's about the same. So in 3 years with imaging instrumentation, you can get the same kind of information. I mean, the same amount of information that if you were using sediment trap for 30 years. But now it's where everything gets complex, because you have a very complex view of the community on the left-hand side, and a complex view of the process of flux on the right side, with two different approaches. And we had to find a way to put that together if you want to understand the process. So usually people, when they face a lot of data, they talk about big data. So I was trying to come up with a definition of the big data, because I'm sure you all have a definition of big data here, but it's not the same for everybody. And so I came up with a definition from that guy that is a professor in psychology and behavioral economics. And to me, it's the best definition of big data. Okay, don't worry, there's nothing out of the, I mean, it's fine. So it's about teenage check. Why? It's because everybody talk about it. Nobody really knows how to do it. Everyone thinks everyone else is doing it. So everyone else is claiming they're doing it. So I had to come up with that because it's halfway to my talk and I needed to wake you up. But so this is a good definition of big data, actually. And please don't tweet it, because my last talk, the only thing that was tweeted was that. But actually, we're not doing big data here. We're working with complex data. It's not exactly the same. It's a lot of data, but basically it's a lot of big matrices, and we play with big matrices. It's not exactly the big data science. So we came up with a method that was already published, actually. It's a method used in system biology to look into network. So if you have the network on the left here, we try to find a way to cluster the network, and it's the WGCNA method here. We could use other method, but that was a standard method that gives you an idea of that could partition a network, a co-occurrence network, based on scale-free methods. So the way the method works, don't worry too much about the detail, but it takes the network, it clusters the network, and link every part of the cluster, every cluster to other parameters, like when you do a PCA, for example. So what was interesting here, if you just take the eukaryote data, the black box on the heat map on the top here, it's the only section of the network that could be correlated as a whole to carbon export, one component of the biological carbon pump. So it starts to be very interesting because instead of having a huge complex view of the community, you can have one bit of it, something that our mind as human can understand. So you take that box and you can open it. So when you open that box, you have a definition of OTUs or here eukaryotes based on metabarcoding data that are linked all together and at the same time correlated to a process or to a variable or whatever. Here, it was correlated to carbon export. And what is interesting here, it's instead of looking at individual bugs, like people have been doing for 30 years, trying to find the usual suspect or the key player, you look at the key players in a community. And it's very interesting because it gives you an idea of the one that matters but are not directly correlated to the process. So what you have here, it's another way of looking at the sub-part of the network. You have the node centrality that gives you an idea of how central the node in that sub-community is. So it means how many connections you have to it. And at the same time, the correlation, the individual correlation of that node to the carbon export. So what you have here, it's a picture of the eukaryotic community linked to the process. And it was very interesting to me because diatoms were there, the usual suspect, but the one that came out as very important based on an additional analysis that we did, so here it's a PLS that extract from that subnetwork only the one that matters if you want to predict the variable. So it's VIPs, it's not for very important plankton, but it's a variable that is important to make a prediction. So if you take only the one that matters in that subgroup, so it's about 100 here, you extract a few that if you use them, if you tag them in the community, you can predict the variability of your measurement of carbon export at the scale of your cruise. And so what was interesting is that Colodaria came out here. And Colodaria, it's a rhizarian. And you find few evidence in the literature that these bugs can matter in terms of carbon export. And you can understand why. It's because you don't have a strong correlation, direct correlation to carbon export, but it's very central to the community. So we did that for the eukaryote, and we did that, and then we went back to the data set and looked at the imaging data, because we were trying to find an explanation why this group of organisms were important. And based on imaging data, what we found is that actually they are very important in plankton in terms of total abundance, but you can't really, I mean, it's not easy to collect them because they are destroyed when you collect them with nets. With imaging instrumentation, the water column is not disturbed, the sample is not disturbed, and so you have a lot of images that you can use, including the fragile plankton. And so that's an additional paper that we did on that, is the distribution of this group. And you can see what you have in blue, it's their latitudinal distribution, and in orange, it's the copepod latitudinal distribution. And between 30 north and 30 south, this group is as significant in terms of total abundance than copepod. So that was the story about the eukaryote, but we wanted to understand a little bit about the prokaryotic, because, I mean, prokaryotes are obviously very important for a lot of biochemical processes, but they're not really well known in terms of the biological pump. And we did the same analysis with the different data set, the prokaryotic one. And what was interesting is that Sequestration came up as one of the bugs that really matters. And finally, we did an additional analysis with the viruses. And to me, that's where it got very interesting because it's an independent data set. It's not based on the same filter or anything. It's a different data set. But the bug that came out as very important in terms of carbon export were the phage of Sequestration for the viruses. So here, everything came together. It's, you have a picture of the community, even though viruses were not thought to be important. It seems that it matters because of their role in the community. And one thing that I want to point out is that if you use few viruses to make a prediction of the variation of carbon export in the ocean, you come out with a variation that is good, that can predict almost 90% of the variation, 89%. And if you take everything in the literature, people, when they try to predict the variation of carbon export, they come up with, when they're very lucky, with 60% of the prediction. So it's a way of thinking about the process. It's a way of thinking about the community. Here, it's a clear idea that the community matters if you want to understand the process. So you need to understand the community, and you need to understand how they interact together. And so the nice way of putting that together, it's also using a graph, but the subgraph, the sub-network of species from virus to eukaryote that are linked to the biological carbon pump. So we did an additional study because we had the eukaryotic component. We could also look at the genes that were important. And that's where for me, it's one of the most interesting aspects of this paper, is that not only using species, you can also use genes, only genes, to understand and predict the process. And here what came up was not one subnetwork, but two subnetworks. And one was primarily related to primary production, and the second one was related to coagulation. So it's again, it makes sense. I mean, you need to produce a lot of material, and this material need to coagulate and get together if they want to sink to make an efficient transfer of carbon to the depths. And so you have to think as this is the first step of designing a probe or a microchip, for example, to tag biochemical process in the ocean. So why do we talk about social network? It's because even though this is about plankton, what we did, it's the same way, it's exactly the same thing that Facebook is doing. So it's building a social network of plankton, building a community that you understand if you want to understand the process. So I mean, Facebook is very good at putting ads on your page, trying to make you buy stuff because you have friends that have been buying the same things. And here it's exactly the same. If you understand well the community that is relating to a process, then it starts to get very interesting because you can learn much more about the process itself. So just a few things. I mean, that was the science they made in Tara, but Tara is not just about science, it's also about communicating the science. And I'm just going to finish with a few examples of the type of communication that we've been doing. The good thing about Tara, it's a group of scientists. It's a lot of scientists, 200 scientists. And it's a foundation, the Tara Foundation. And we really work together. We're not trying to do the work of the other group, but we work together to put the science out there. So we communicate to scientists, we communicate to journalists, and we communicate to the foundation, and the foundation then communicate to journalists and stakeholders and politicians. So that's some of the communication that we've been doing. We had a lot of press, a lot of media. It's more than, I think, 2,000 different newspapers now that have been talking about the expedition. We had books. And what we try to do also is to teach to the young. So every time we go at sea, we try to stop in harbor and try to get classes on board to talk to kids about the science that we are doing with simple words. It's always very interesting because, I mean, when you talk to kids, you try to talk to everybody. You try to put simple words on very complex science that you try to do. And it's more challenging than talking to you, actually. But it's part of communicating the science and try to inform the public about the science that we're doing. And we're doing that in close collaboration with the foundation. We have a lot of clips. If you go on YouTube, you can find also all the clips that the Tara Foundation has been doing. So you can learn a lot of things about the science that is done on board, but you also have a lot of communication tools. And one thing that was interesting to me is that the plankton imaging that we've been doing in Tara, and mostly Christian Sardet, that is one of the PI of the group, have been reused in a song, and it's the Mantare song. I don't know if you heard about it, but it has more than 2 million views now on YouTube. So it's just a way of communicating. It's another way of communicating about the science that we are doing. We also developed tools to try to talk to the kids. And Fabrice Not has been working with people from his group to design cards so the kid can play with it and learn at the same time as they play. It's the Planktomania project. You can go on the web and get information about it. And it's a nice way also to talk to kids and to make them play with interesting information. And he also developed movies of augmented reality movies that you can use to learn, to teach stuff in class. And Christian has been really active trying to communicate about plankton also. And he has a lot of books in different, in at least 4 different languages that have been really well sold actually. And people have been really, it's interesting to see that. I mean, when I started to work in the project 10 years ago, I didn't think that people were really interested about plankton. But when you see the echo, when you go out there, people now really know about plankton, even the people that didn't really care about the ocean before. So it's a way to communicate. And finally, the thing that we try to do, it's also with the foundation. It's not the scientists directly, but they try to go to policy. And so you have people in the Tara Foundation that are doing that job. And so one thing that they've been doing after the expedition is to set up an exhibit in Paris at the COP21 and really to inform the policymaker basically about the science that we are doing and why plankton is important in the ocean, why we should study it, why we should study the ocean, just the ocean, because that was one of the big outcomes of the COP21. So everything, I mean, we are a big group of people from the science side and from the mostly like NGOs and stuff like that, and we really try to work together to communicate based on the science that we do to the stakeholder and the policymaker. So, and it works very well when everybody is doing his job. So I just want to thank a few people, especially the one that really inspired me to do this research. So Gabi Gorski is on the left. I didn't talk about Gabi Gorski, but Gabi was at the beginning of the expedition. He's one of the main founders of the expedition with Eric. Eric was the sea biologist and Gabi was the oceanographer, and that's because they talked to each other that they decided to do the cruise, and they decided the way to do the cruise itself. Dave Carl, that I've been working with when I was in postdoc in Hawaii, that really gave me the idea of working with omics and in microbiology, even though I'm not a biologist. Chris Boller and finally Eric Santi, that are 2 of the main PI of Tara that are always very inspiring when you talk to them. Marc Pichral, that is on the left, that is bottom left, that is the engineer in our group that is developing the instrument. And for those of you that have good engineers, you know that you can't do research if you don't have engineers. And the 3 people on the right, it's Lucie Bittner, Samuel Chaffron, and Damien Eveillard, that are people that have been stuck on the boat with me and are doing very different science that I'm doing. Lucie, she's working in evolution. Damien, Sam is working in microbiology. He's been working mainly in the gut, human gut microbiome. And Damien, that is a computer scientist. And because of these 3 guys there, we came up with the study that we did. It come back to the first plenary talk that we had here. It's mostly about people and the trust between each other and the way we communicate. I mean, we've been stuck on the boat. Obviously, it helps to communicate. But we've been stuck on the boat together for a month, trying to get to the same level of science, even though we didn't have the same background. And because of that, we managed to come up with a new idea to analyze a process that was of interest for all of us. But it's not my science or their science. It's now a bit of our way of thinking about the science that we want to do together. And that's not the only people that I have to thank, obviously, because the paper that I talk about, it's about like 40 people. So everybody has been working really hard on this project, and they all have a different role, but it's because we're all working together that we managed to do the science that we do. And finally, I just want to thank ICES, because that's why I'm here. I'm here today, and it's been a great meeting, even though I'm a bit of a newcomer here. But I hope that with that talk, you understand a bit the research we've been doing in Tara, and why studying the full community can give you a better idea on the process that you want to try to understand. So with that, I thank you for your attention, and if you have questions, I'll be happy to discuss with you now or after the talk.
I'll leave you to grab a drink for a moment. Thank you very much for that excellent talk, very inspiring, very informative, but also I like the approach about looking at data and trying to convince people to think about data. It's really good, but equally the multidisciplinary nature of the work is really, really interesting. And also thank you for actually being so good with timing as well. So we have plenty of time for questions. Anybody has any questions for Lionel? I can't really see very well, so anybody has any questions or any comments? Everybody over there? Do you have a mic? Sorry.
Yeah, don't be shy. It can be about anything, even the life on the boat.
Thank you for an inspiring talk. What are the implications of your new findings for the global climate models that predict climate change? How different are your findings from what are coded into those models?
Well, I'm not a climate modeler, obviously, but I don't see, I mean, it's not a limitation per se. The limitation is mostly based on the data, because omics data, for now, there is no way they can get into climate model. So that's one of the limitation, technical limitation. And the implication of the research that we've been doing is such that if you just try to model one bug or the one that you think matters based on, I don't know, its physiological characteristic or whatever, like in most of the model that you have now, it's based on macrophytoplankton or picophytoplankton. It's boxes mainly, even though you have like functional types or stuff like that. But it doesn't give you a real idea of how the community is functioning. And so to me, that's the main limitation, because you can still manage to model that bug and try to inform on the functioning on that bug related to climate research, how it's going to impact or reply to climate variation. But at the same time, you don't know what's going to be the interaction of that bug with the rest. And everything is about the interaction here. It's not about the bug itself. So that's one of the big challenges. Are we going to put that into climate model? I'm not sure yet. But you have to think about going in that direction, because interaction shape the community, and interaction also shape the adaptation of the community to a change. So that's why we have to keep in mind when we do this kind of study, because it's where the change is going to happen. It's not per se on the bug itself. You can have changes of bugs that's still going to have the same function in the community, and the feedback on the climate will be exactly the same. So that's what we have to keep in mind, and it's a big challenge, I think, for the full community, the scientific community, for what is coming.
Your presentation was lovely, and the amount of data you showed is unbelievably astonishing, as you say. And so my question is a bit off, because you just said it's not about the bug, but I wanted to ask about the bug. One of the numbers that was so impressive was your 130,000 OTUs that you turned up. And I wonder, have you thought about the relationship between those OTUs and the species diversity that you might be seeing? And what do you think about that in terms of how you can translate that?
Well, that's a big, it's a big challenge. So I'm not a molecular biologist or a biologist itself. I'm mostly an oceanographer. So I'm trying to think about data, mostly. And to me, OTUs is just an abstraction of the biology. So I don't think about species itself and the diversity of species, but the diversity of OTUs is still an information. How it relates to real diversity, to me, we're going to have to combine data sets. Imaging data can give you an idea about the diversity, some kind of diversity, and at the same time, it's quantitative. And omics data are mostly semi-quantitative or qualitative. You have relative abundance, stuff like that. And it's about OTUs that are an abstraction of species. So combining the two data sets, it's where we're going to get a good idea of the diversity, I think. but it's going to take a bit of time because it's not simple. It's not a simple task at all. So that's where I would go, but again, I'm not a biologist, so I can't really answer about the OTU diversity versus the real diversity.
Yes, right here. Thank you very much again it was a really awesome presentation. My question is related to what was the most shocking thing that you saw on the expedition? One thing that I've been thinking about is the prevalence of marine litter when you're taking such long transects, and if there were perhaps even pieces of plastic that you can find at the micro level when looking at all your different instruments that perhaps showed up, or were there other maybe things that you didn't expect to see?
Can you just repeat the question? It's just about marine litters, or?
Well, that's, for example, did you see a lot of marine litter? Other people have reported that when they do these large expeditions, that's one thing that is shocking. But when you're using so many different instruments, if you found things that you didn't expect to see, for example?
I was not going with thinking of finding anything special. So, but about plastics, I mean, plastics are everywhere. We had manta trawling also during the cruise, and people are working on the plastic side. So I'm not really into that science, so I'm not sure what kind of result they are coming up with. But plastic was almost in all sample that we got. Microplastic, I'm not talking about big chunk of plastic drifting. But yeah, there is one of the cruise, Tara expedition. So Tara is divided in different expeditions. There is here it's Tara Ocean and Tara Polar Circle, and there was the Tara Mediterranean Sea expedition that was mostly about plastic. It was 3 years ago, and so there is a consortium working on that and trying to come up with an idea of the distribution of plastic and the impact of plastic on plankton communities at the scale of the Mediterranean Sea. So probably more results are coming on that topic, but I can't comment much more than that.
This is extremely impressive, and what has impressed me the most, I think, is this amount of OTUs that have not been identified. So it's about more than half, perhaps, that we don't know what is in the sample. So, yeah. So is that out of your analysis?
So it depends. It depends on the analysis that we're doing. For some, you don't, because you don't have any idea of what they are, you can't use them. But we have people, mostly in bioinformatics and biology, working on the unknown and the unknown on the database to try to put a name on it first, and also to look at their distribution. Even though they are known or unknown, you can do analysis on this, or include that in the full analysis also. So it's not just about unknown taxa, it's also about unknown genes, because if you remember one of the last slides about the genes related to the biological carbon pump, it was about 70% that were unknown. And so that's obviously a big unknown, but with this kind of analysis, at least you can extract a fraction of the unknown that matters, and then tag these ones to try to understand what they are. And so that's also one way of thinking about the data set. Even though you don't know what they're doing or you don't know what they are, at least you can start to have a small fraction of the unknown that you can use and try to understand.
Hi, I'm just curious where the boat is now and what the next expedition might be and focusing on.
So the boat is in an expedition right now, and they're doing, it's mostly based on coral research. They are in the Pacific and trying to look at the microbiome associated to corals at the scale of the Pacific.
Has there been any attempt to sort of link up with the Carlsberg, about a century ago, Carlsberg with Schmidt Johann Schmidt led the expedition around the globe, of course, traditional methodology. But they've been working on, ever since, 100 years now, of identifying the samples and archiving. So you probably could have a 100-year comparison for at least with the traditional sampling, what was there, compared to the kinds of things you're seeing. And they did do plankton around the world in the project. Is there any link up with the Carlsberg?
I have no idea.
Something to think about, to reflect on.
Yeah.
Any more questions or comments? No? Okay. I've got a question actually. So what's next? Because it seems that the expedition is looked at all sorts of different angles, so from the outreach to policy to the science, but any specific topics that you're going to concentrate on?
Well,
any priorities at the moment that you are thinking? You talked about evolution, you talked about different angles.
yeah, so one aspect is actually about the climate research. We really, I mean, that's one of the things that I'm trying to develop, is try to see how we can put genes on the map of climate research. So that's going to take some time, but it takes also a lot of different people, from the climate to the computer science, through the omics, through the biology, to try to see how we're going to be able to do that. So it's one objective that is on the technology side, yeah, but at the same time it has a big implication for modeling the system in the context of climate change.
So several aspects, I suppose, of the work. Yeah.
Yeah.
Very good. Sorry, anybody has any more comments or questions? Sorry, yep, over there. Sorry, hang on.
Thanks for a very interesting talk. I was just wondering if any of your genetic material that you found you can associate with macrofauna, fish, or marine mammals, for example?
No, we haven't been working on that yet. We're going to try to look at fish tags in the data sets, but for now it's not done and nobody's really working on that in the group. We have a postdoc that is, or a PhD, no, it's a PhD student, not a postdoc, that has a focus on that, a small focus on that. So we'll see what's coming out, but for now it's completely an open question.
Hey, that was really impressive amount of science that you got done, and it's also impressive on the budget that you had. So I was wondering if you had any, if you could translate how you were able to get so much science done, if there's a lesson that we can learn with the other science that we're all doing.
Can you say it again? About the budget?
It's amazing how much science you got done, yeah, on the budget that you had.
Okay.
And I was wondering if there's any lessons there for how the rest of us are doing science.
Yeah, for the budget, well, sure, it's 9 million, it's significant, but at least for the science that we've been trying to do, it's not that much, but it doesn't include the people the French salary, because we're lucky, we don't have to look funding to get for our own salary. We need funding for postdoc and student, but for the researcher, there is no need. So that's one at least positive aspect. And the thing about a limited budget, it's that you have to do the most of it. So we really try to get together as much as we can and work on the data set together, even though the budget is not that, the budget is not extensive. We try to do the science that we can with it. For the sequencing, it was a big chunk of the funding, and we've been working with a Genoscope in France. It's one of our sequencing platform, basically, to try to get the most data out of the funding that we had. And so, yeah, we are in constant search for funding, additional funding, to try to bring new folks in the group. I mean, a new PhD student, new postdocs, or even new outside researcher, because there is a lot we can do with the data set that we are not able to do with the funding that we have. So that's, yeah, funding is always a limitation.
moment? Anybody? Any more questions? No? Okay, well, once again, we would like to thank you because I know you're extremely busy and I know that you have a lot of things planned afterwards, but before you go, as you say, you're a newcomer to the ICES community, and we for sure welcome you, but also would like to keep you. So I have a series of little gifts for you to take home as well as a token of appreciation, but also to say thank you for a really inspiring talk. Thank you for coming to share your science, and equally, we're really delighted to have you. So once again, thank you, and I'll give you some gifts, and
thank you.
if you clap again, that would be great.